Now let me concentrate on cytoplasmic inclusions. They're the non-living substances of the cell. They're not bound by a membrane, and they include lipid droplets, zymogen granules, glycogen granules that we see in the digestive glands, and we also often see pigment granules in some cells. And then another inclusion is going to be aggregations of secretory granules, but they're membrane bound. Here's a section through the adrenal cortex, and here are large cells with a large dark nucleus, and their cytoplasm is full of little round circular white structures. They're lipid droplets. They dominate the cell, and that's because these particular cells are making steroid, and the lipid droplets are cholesterol stores. They're the precursors for all the production of the steroid hormone that the cell is producing. They appear clear in the cytoplasm because, being a lipid component, that lipid component is lost during the processing of tissue, and therefore they just appear as a ghostly white structure within the cell. Glycogen appears to look like soot scattered over the cell cytoplasm in this electron micrograph inside this liver cell or hepatocyte. That glycogen is stored in the hepatocytes and is the energy source we use, or one of the energy sources we use in our bodies. You don't normally see it in a normal histological section. You need a special stain to see glycogen, and I'll talk about staining and what stains indicate in another lecture in this histology of cells and basic tissues course. Here's a rather high magnification image of skin, and you see here a curved line of cells and nuclei, and this brown pigment is melanin. Melanin protects the very basal layers of our epidermis from any damage from ultraviolet light. These basal cells are constantly dividing to give rise to cell layers of the epidermis. The melanin protects those dividing cells from any DNA damage that may be caused by excessive ultraviolet light. There is also neuromelanin, which is a similar product that we have in the retina of our eye. When lysosomes break down components that I spoke about earlier, the breakdown products remain inside the cell as residual bodies or age pigment, and it's got a yellow, orange stain or colour to it. We call it lipofuscin, and there is some in these particular cells here. Now, you can also see a similar pigment to lipofuscin in these cells here. It's called hemosiderin. Hemosiderin is the residual components of the breakdown of hemoglobin. Red blood cells only live for about 120 days, and when they pass through the spleen, the old ones can't squeeze through all the network of barriers the spleen puts up to test whether these red blood cells can squeeze through. The old ones can't, so they get caught in the network of fibres or networks of reticular fibres that are set up in the spleen, and then macrophages come along and break them down. They break them down and reuse part of the components of hemoglobin. The body is very much into recycling, but the residual amount you see here in the cytoplasm of macrophages within the spleen as an orangey browny component is hemosiderin. Secretory granules are membrane bound, and they're produced by cells, particularly protein secreting cells, in all our glands that secrete digestive enzymes, saliva and so forth. And in this image, you can see in these groups of cells very pink staining little tiny granules. They're secretory granules, and they will pass to the cell surface and then out through the lumen and then pass through a duct system to the part of the body where they're going to be utilised.